here is a good intro to this slide, which was the postdoc office circa 2004. I can't remember the exact date, but I do think there was an election coming up, and that plaque read something very different than Happy Doug Day at the time. So I'm not sure when I first met Doug. It was probably when I interviewed to be a postdoc in that room with people like Matt Strain, who's in the audience here, and Davey in the back, Stephanie Rout, Kirsten Kolsch, and Kevin Morris. But I do remember when Doug first noticed me, and I think it was probably right around this time, Jacques, who's also here today, had left to go back to Canada. And as a somewhat cocky postdoc, I wandered into Doug's office and said, if I were to get a grant, I'd like to take over the Genomics Core, have my own office, and be called something other than a postdoc. And grant funding, as you know, is very difficult, and I think Doug smiled and I went away. And I came back actually probably a year later after finally picking off a grant, and I said, I got my grant, can I now have my own office, get out of the postdoc pool, and take over the Genomics Core? In hindsight, I should have probably added a lot more things into that list. But Doug looked at me and said, is that what we agreed to? And I said, absolutely. And a man of his word, all of those things happened. So that's, I think my earliest recollection. Yep, so again, I've bounced around a bit in this half a career so far. Hopefully I'll stay put and be more productive, and Davey can introduce me a little better next time. Uh-oh, what did I do? No. And so the really sunniest years of my career, both professionally and with the weather, were here in San Diego at the Center for AIDS Research, running the Genomics Core, meeting a lot of people in the room, and doing some great science. Again, I'm at Merck now, but won't be discussing any of that research. So any of you that have had kids going through 4th grade probably know what an acrostic poem is. I had no idea what it was until my girls started drawing them up on the whiteboard, left, right, and center. And so these are those poems that you get to go down and make words from another word. So I thought I would structure my talk and in honor of Doug being an Old English major around an acrostic poem. So D is for determined, which may not resonate with you all of a sudden until I put this phrase up. And I can judge by the chuckles that probably everybody in this room has heard this phrase at one point. It is a golden rule I took away with me from UCSD and has got me in far less trouble than it has actually advanced science. So I'm going to tell you about a reverse vaccinology project that I did that basically embodied this concept where we couldn't get funding, but we just went ahead and did it anyway. O is for opportunity, and that speaks to Doug being a great mentor. He introduced me to this collaboratory of AIDS researchers for eradication, which is a real highlight of my career, and I'll tell you a little bit about the work we did in that group. Understandable, so when I was at the CFAR here, we had work in progress and journal club every week. I still see through the announcements through Facebook that you're continuing to do that. That was essential for me to understand some real complexities of HIV biology. And so I've always tried to carry that forward. And I'm going to tell you a little bit about a complicated head and neck cancer study that we made understandable when I was at the University of Southampton. And G, going back to the English major theme, is for grammar. So Doug was always a stickler for grammar. I see his daughter nodding, so I'm sure that was at work and at home. And I'll give you a little example of that to finish with. So D for determined, I was going to start off with this reverse vaccinology project. So the goal here is, I see Theo in the room too, we worked on some of these projects with a fungal pathogen, is to take a bacterial pathogen genome and identify protective antigens straight out of the genome that could be incorporated into subunit vaccines. And I had a master's student from SDSU who started this work at the CFAR, and then it was taken up by a PhD student in Southampton. And reverse vaccinology has been around for a while. The real premise of it was supposed to speed up vaccine design. I don't think that's really true because you still have to get into animal testing and you still have to go through the FDA regulations. So I see reverse vaccinology as just another arm to subunit vaccine design that relies on a computational approach. So why should we be interested in reverse vaccinology? So Rino Rapuli's lab first spearheaded this effort, and in fact, they got all the way to a clinically improved vaccine for MenB called Bexsero, which is currently on the market. But again, it took them about 10, 13 years, so it was sort of the standard vaccine timeline. So our motivation for this study was that original method was simply a filtering approach, like peeling an onion. They would go into a bacterial genome and they would throw out any intracellular proteins and only keep the extracellular ones. They would only keep proteins that had a single transmembrane domain, and so they were throwing out a lot of information. And the pie chart on the right shows you if you go into the literature and you look for these protective antigens, then they're actually distributed all over the bacterial cell and not just on the surface. So we had two main goals: to reduce the number of protective antigens requiring laboratory validation, and to identify all protective antigens regardless of where they were in the bacterial cell. And we were going to use machine learning approaches to do that. So here's the pipeline that we put together. When you're applying machine learning, it's often called artificial intelligence in today's world, but it's really machine learning. You need a training data set. So we went into the literature and we found all the cases where someone had taken a bacterial protein, injected it into a mouse, raised an immune response, and then that immune response protected that mouse from challenge with the bacterial pathogen. And that was our positive training data. The negative training data was just randomly selected proteins from the same bacterial genomes. We then annotated that protein data with protein annotation tools, and we trained a support vector machine classifier to basically recognize a protective antigen or vaccine candidate from a non-antigen. And you can then point that classifier at any bacterial genome, the protein coding genome, and you can rank all those proteins for their likelihood of being a vaccine candidate. So obviously there's some questions that follow that pipeline. Do we need to use this fancy machine learning methodology? Well, this plot shows that as you increase these features, these annotation features that we annotated the data with, the classification accuracy goes up, and then it starts to tail off because the more features you add in, you're adding in a bit of noise to the classifier. But on the right, you can see that the support vector machine or the machine learning approach outperformed these other regression methods quite effectively, had much higher accuracies. And then, are we doing any better than chance? So the other thing you can do is you can scramble the labels of your training data. So you can start calling non-antigens protective antigens or vaccine candidates, and vaccine candidates non-antigens, and that should have no signal. And you can see that along the diagonal line here. This is a receiver operator curve, which is how we evaluate the accuracy of a machine learning classifier. And if you're doing no better than chance, this curve goes along the perpendicular line. Our actual classifier, trained on the real data, has that nice black line, which meant that we were doing better than chance. If the classifier was doing perfect, i.e. it was ranking all of the vaccine candidates that we know about at the top of the list and all of the non-antigens at the bottom of the list, you'd get this sort of square going up the vertical axis and straight out the horizontal axis. So we're somewhere in the middle, but it shows that these classifying approaches had some signal. And then finally, we invented this new statistic called recall, where if you're ranking a bacterial genome, all the proteins in that genome for their likelihood of being a vaccine candidate, where do the known candidates or the known ones from the literature fall in that ranked list? You would hope near the top if your classification approach was doing a good job, and we used a hypergeometric chess to prove that significantly. So this led to quite a few publications. I had a quick look before I flew out, and it was actually quite gratifying that Rino Rapuli, who originally got the Bexsero vaccine approved for MenB, did a benchmarking exercise this year for all of these approaches to reverse vaccinology, and he called out our method as being the best method of all the approaches that exist out there today. So I think we did something right. All right, so O was for opportunity, and this was the collaboratory of AIDS researchers for eradication. This was a large academic collaboratory, the first in the country really tasked at tackling issues around curing HIV and eradicating the virus. Our small part in this collaboratory was to try and determine the off-target effects of a compound called SAHA or vorinostat in primary CD4 T cells. And Corey, who actually works with me at Merck today, was my first graduate student who helped with this work. And Nadia, who many of you know in the room, this constituted her postdoc studies. So this slide is probably somewhat redundant to this room, but the shock and kill strategy for HIV cure was this concept that the virus is hiding out in cells and we don't know where those cells are, and that we need to activate those cells to start producing virus so that the immune system can see them and kill them. And one of the compounds that we use for activating is this vorinostat or SAHA that I told you about. So this was one of our first forays into sort of a more complicated systems biology approach, where we basically did both a transcriptomics and a proteomics analysis. So we treated primary CD4 T cells with the compound SAHA, and then we did a mass spec on-targeted analysis, iTrack proteomics, and an Illumina B-trip transcriptomic. And then you can identify the differentially expressed genes, differentially expressed proteins between treated and untreated with this drug. We could also get at differentially phosphorylated proteins and differentially acetylated proteins. So that is a whole mess of data, and we scratched our heads about how we were going to make any sense of it. What we ended up using is a protein interaction network. So there are companies out there that have curated protein-protein interactions, and we can push our differentially expressed genes and differentially expressed proteins onto these networks. Corey's genius was carving up each of these nodes, which represents a protein, into whether there was a signal for differential expression at the transcriptomic level or at the protein level, or if they were differentially phosphorylated or acetylated. Now that did result in a hairball, which is often, my hairball comes back, the criticism of these types of studies. But by scanning through here with the eye, it was very easy to pick out one single node, HMGA1. And so this particular protein was upregulated at the protein level, upregulated at the transcript mRNA level, and also showed increased acetylation. So why is that important? So it turned out that HMGA1 is a transcription type factor that binds to TAR at the beginning of the HIV sequence and actually represses transcription and activation of HIV. So if you remember from the beginning, I was telling you we were trying to use this compound SAHA as a compound to activate HIV, and one of the off-target effects, it's also activating a transcription factor that represses the transcription of, or that activation of HIV. So obviously this compound does not have an ideal off-target effect, and if we had run this screen a lot earlier, we probably would have moved away from this compound in the collaboratory faster. So this led to sort of lots of publications at the transcriptomic level. This was the work I just told you about. And then we also did some nice dose response work. I think what's really gratifying is to see that Nadia has continued with this work and has moved on to other compounds in the same class that have shown better efficacy, and she's examining some of these hypotheses around HMGA1. Okay, so my third vignette was U for understandable. And I want to tell you just a little bit about the head and neck cancer studies we did when I moved to the University of Southampton. So having been at the CFAR for about 10 years, and paying attention in journal clubs and work in progress, I knew that virus was bad. And so I took that away with me, and I went and started collaborating with this oncologist, Christian Ottensmeyer, and my postdoc at the time, Jean-Min Wu. And it turns out that viruses can also be good. So in this case, you're looking at a survival plot of people with head and neck cancer. So U was for understandable. What this Kaplan-Meier plot tells you is if you have an HPV-driven tumor, so human papillomavirus, and you have a lot of lymphocytes infiltrating the tumor, you actually do pretty well. You survive longer. Whereas if you don't have any virus in your tumor and you don't have these lymphocytes infiltrating the tumor, then you sort of have a poor overall survival, and patients die faster. So sort of the overall hypothesis is that there's a virus in the tumor which is driving the patient's immune system towards the tumor and is leading to eradication of that tumor. So the question that our oncology colleagues had was how do we better understand this tumor environment in HPV-positive tumors that's driving these better outcomes? So they set up this incredible clinical sampling procedure where they were able to get biopsies straight from the patient before surgery. They were immediately frozen. We got the nucleic acid material out of the samples. They're actually sent back here to a collaborator who did the sequencing just down the road. And then the data came back to us, and Jean-Min did the analysis to figure out which genes are being turned on in these HPV-positive tumors compared to HPV-negative tumors. And it was a fairly small study. We only had 10 subjects who had the virus-driven tumor and 13 that had the non-virus-driven tumors. So when we identified that list of differentially expressed genes, one thing you can do to try and make sense of that list is map them onto these ontology terms, which are groupings of genes in similar functions. And immediately it seemed to make sense, right? There were a lot of sort of lymphocyte and immune cell terms here, which sort of makes sense because the HPV-positive virus-driven tumors seem to be recruiting these cell types. However, this is where the understandable part came in, because we sort of did the thought experiment around this. If we have an HPV-positive tumor and it has more of these lymphocytes, then when we compare it back to an HPV-negative tumor, is our signal for differential expression being driven just by the fact that we have more of these lymphocytes in the HPV-positive case? So you can imagine if this lymphocyte's a CD4 and you have 100 over here and only 1 over here, any gene, even the CD4 marker itself, would show up as differentially expressed just because the cell numbers are different. And so that's what we sort of called numerical differences. But what we were after was if we pulled one cell, one lymphocyte out of the HPV-positive case and compared it to one cell at the HPV-negative case, what was actually going on a cell-to-cell basis that was different. And this is a whole field called deconvolution, which is trying to take this kind of data and adjust it so you're really comparing the same number of lymphocytes across these two disease groups. So I won't go into details here. We used a technique called FAME, which basically took gene expression markers of cell subsets, and we were able to estimate, using the gene expression, how many cells we thought were in each of these tumors. And encouragingly, we could see lymphocytes like T cells and B cells were estimated to be, of course, higher in the HPV virus-driven tumors than the negative tumors. We used that information to correct the data. So originally, we had sort of thousands of differentially expressed genes. When we corrected sort of for a cell-to-cell comparison, we lost a lot of that differential expression. And that was the original ontology analysis I showed you. A lot of that disappeared and got us down to the true biological differences between HPV-positive and negative tumors. And when we went back and we had a look at these corrected differentially expressed genes, and we went back and had a look at what cell types they were related to, our oncology colleagues kept beating us over the head and telling us it was all about T cells. But the signal that emerged from the data was that, in fact, the differentially expressed genes that survived correction were mostly related to B cells. And so it seemed like the B cells were doing something different in HPV-positive tumors than in HPV-negative tumors. And when we pulled some of these B cell markers out and plotted them in the TCGA, which is a big cancer repository for data, we could see that they were actually prognostic for survival if you stratified patients by high and low expression of these markers. And so we published this while I was at Southampton. And of course, that was a very sort of complex way to answer this question. These days, new technologies come out called single-cell RNA-seq, where you could actually just analyze. I see Jack nodding, yeah. Glad I avoided that question. So you could just do single-cell RNA-seq analysis and get to this single-cell question. And in fact, that's being done. And again, digging into the literature before coming out, it appears that other groups have done single-cell analysis in this cancer type, and they have shown that B cells are really important for driving good survival in head and neck cancer. So that brings me to G for grammar. And I thought this was going to be hard. I thought I was going to have to search my old hard drive. But in about 5 minutes, I found an old manuscript that had been edited by Doug. I really need to Doug was a fantastic collaborator. Any manuscript, you ping him, he reads it in detail. There's always some really good revisions in here. This isn't a really good revision. This is my continued failing to realize that data is plural. And so I could bore you for the next hour showing you manuscript after manuscript with this same correction. What I'm really got surprised at was there was always this sort of non-aggressive comment, just a simple deletion and a change. I think if it had been me, there would have been some more sarcastic comments in the margins going forward. But Doug, thank you for all those corrections. And that was pretty much all I had. So this was a CFAR AIDS walk that they run in Balboa Park. Doug always came down to join the team, and it was great walking around Balboa Park with him. I have some very fond memories of San Diego. This was the leaving party that the group organized for me. We had some nice cake. It was a happy and a sad day at the same time. My twin girls were born here, and so we have very fond memories of hanging out with Davey and others. And then I've made lifelong friends here who are still here today, and they're still talking to me. Well, we'll see how we do after today, right? All right, fair enough. Thank you very much.